All right, with that as our background, we kind of see, if you watch part one, you kind of see how the autonomic nervous system is laid out. What we want to do now is we want to explore a little bit more about the physical anatomy, the structure of how this stuff comes to be. So let's look at a couple of different pictures. Here we've got a picture of the CNS, of course, the brain and spinal cord. Anatomically, where are the sympathetics and parasympathetics exiting the CNS from? Well, they have very specific regions. If we're thinking about the parasympathetic system, 95% of the parasympathetic system exits from the brainstem. We see this in, for example, cranial nerves as a great example. I'm thinking cranial nerve 10, the vagus nerve. It's loaded with parasympathetic neurons. The other 5% is at the complete opposite end. So down in the sacral area of the spinal cord, this is the last 5% of the PNS, and these neurons that are exiting here innervate the pelvic floor. I'm thinking reproductive organs, bladder, that kind of stuff. For the sympathetic system, they exit the spinal cord in this region here from T1 to L3. Those are talking about vertebrae, the thoracic and the lumbar vertebrae. This is where I get all of my sympathetics. And I've got another video in the next lecture detailing the different routes that these take in particular. So now that we have that broadly matched up, we understand where these neurons are originating from. It's important to kind of compare and contrast the sympathetic and the parasympathetic systems, and even compare that to the somatic system and how we might find similarities or differences, right? Just a brief recap. If we're looking at somatic, right, skeletal muscle, we have a lower motor neuron that originates from the spine and the ventral horn, and that single motor neuron, myelinated, goes to and innervates a nerve fiber, a muscle fiber. In contrast to that, both the parasympathetic and the sympathetic systems are two neuron systems. So let's take a look at that. We've got two systems shown here, and by default, which one is which? While there are exceptions to the rule, we can with confidence state that this bottom one is a sympathetic system, whereas the top one is parasympathetic. How do we know? Well, it has everything to do with the length of the pre-ganglionic. They're like, what does that even mean? So let's take a look at these and see if we can determine what's going on. First of all, you'll recognize that this is a two-neuron system. The first neuron we call the preganglionic neuron, whereas the second neuron we call a post-ganglionic. Now, remember what a ganglion is, right? A ganglion, ganglia for plural, this is where the soma or the cell body of a neuron is located. And what that means is that this is typically where synapses occur. And so we can draw a circle here. This is going to be the ganglion. Now we have a better understanding of what it means to be preganglionic and postganglionic. As we compare these two, recognize the preganglionic is myelinated, right? We can see the myelin, whereas the postganglionic is not. This is true for both parasympathetic and sympathetic systems. How else can we compare these? Well, let's look at the neurotransmitters of the ganglia. The preganglionic neuron in the parasympathetic system releases a neurotransmitter that you're familiar with. This is acetylcholine. The receptor across the synaptic cleft on the postganglionic neuron is a nicotinic N2 receptor. We talked about N1 receptors in skeletal muscle. This is the N2. It's on the neuron. So it's a very, very similar system to what we have with the neuromuscular junction. We're releasing acetylcholine. We're binding to ligand-gated sodium channels. That's exactly what this N2 nicotinic receptor is. It's a ligand-gated cation channel. And that will always, always, always lead to an EPSP because it's an ionotropic cation receptor. Let's compare that to the sympathetic system. Well, guess what? It's the same thing. We're releasing acetylcholine. That acetylcholine is binding to N2 nicotinic receptors, and it's always delivering this EPSP. What about the postganglionic? What's it doing? This is where we start to see a few differences. In the parasympathetic system, the neurotransmitter being released, again, is acetylcholine. But as we look at our target tissue, whether it be the lung or the heart or whatever, we're going to move away from this idea of an ionotropic channel, a ligand-gated channel, and look now at a metabotropic receptor. As we stated before, these are G-protein coupled receptors. These are 7-pass transmembrane receptors. We'll draw it like this. It's a G-protein coupled receptor. And for the parasympathetics, these are called muscarinic receptors. Now, one thing from the previous movie, or the previous video that I wanted to remind you about, we talked about this idea of a stimulatory or an excitatory versus an inhibitory response, right? What can I do to get the smooth muscle to constrict or to contract or to relax? The signal is always going to be acetylcholine. The question now is, how can I get that acetylcholine to do one thing or the other? And that totally depends on what muscarinic receptor is present. We have 5 different types. We have M1 through M5. And the rule of thumb, it's not always the case, but you're going to be right about 80% of the time if you follow this rule. The odds, in other words, 1, 3, and 5, the odds tend to be stimulatory, where the evens, 2 and 4, tend to be inhibitory. And we'll give an example of that in just a minute, just to kind of polish off what that actually means. Let's go back now and look at the sympathetic system really fast. This is where we start to see some interesting differences. The post-ganglionic in the sympathetic system does not release acetylcholine. The neurotransmitter here released is norepinephrine, also called noradrenaline. It's also binding to G-protein coupled receptors on the target tissue, but these have a different name. These are called adrenergic receptors. It's not quite as clean as 1, 2, 3, 4, and 5. We actually have 2 classes of adrenergic receptors. We've got alpha and we've got beta. In the alpha class, we have 2. We have alpha 1 and alpha 2. And in the beta class, we have beta 1, beta 2, and beta 3. The difference here is actually really, really interesting. The betas tend to respond better to epinephrine, whereas the alphas tend to respond better to norepinephrine. Now, this is interesting because we just said that the post-ganglionic in the sympathetic system is delivering norepinephrine, and that's true. So whenever we're looking at a tissue-specific response, it's going to be norepinephrine. But remember, fight or flight isn't always tissue-specific. Sometimes this is a full-on adrenaline response. So we get that adrenaline rush. Adrenaline is released from the adrenal gland sitting on top of the kidneys, and it gets into the blood. And so here's our blood vessel, and it's carrying epinephrine. Epinephrine can also bind to that receptor, but the affinity that that receptor has compared to norepinephrine will be different depending on whether it's an alpha or beta. Not super important, but interesting to think about for sure. We also have to address the fact that some of these are going to be stimulatory and some of them are going to be inhibitory. Luckily, the same rules for muscarinics apply to these adrenergics, where the odds tend to be stimulatory and the evens tend to be inhibitory. All right, let's see if we can wrap this up in a meaningful sort of way. Let's visit one of my favorite organs, the bladder. Okay, here's the bladder. Let's think about how we control the volume of liquid that's in this thing. Of course, through the first years of life, you're trying to figure out how to exercise the skeletal muscle. You can see the little boy doing the potty dance, because down here, I've got the external urethral sphincter. Sphincter, of course, is just a round muscle, so when I contract it, it squeezes, and once I figure out how to contract this muscle, that's going to not allow me to void the bladder. This is skeletal muscle. And so going back to the neuromuscular junction, we know we've got an alpha motor neuron coming in and innervating this skeletal muscle. We know that this is acetylcholine being released. We know that it's binding to N1 nicotinic receptors, and this is going to lead to muscle contraction, allowing me to squeeze this and close the door to prevent the bladder from being voided. Okay, so where does the autonomic system come into play? Well, let's look at a couple more muscle types. Number 1, we have here the internal urethral sphincter. When constricted, that's going to prevent me from voiding the bladder. But we also have the detrusor muscle. The detrusor muscle is smooth muscle within the cell or within the bladder wall itself. And I hope you can appreciate, if I want to void the bladder, I need to squeeze the detrusor muscle. So right now I'm trying to make this video and I'm thinking I don't want to have to go to the bathroom. And so this is kind of a sympathetic response for me. I'm nervous. I want to make sure I get it right. So let's follow the sympathetics. I'm just going to look at the post-ganglionic neuron. We'll do this in purple. And that post-ganglionic neuron is going to come in and it's going to branch. And some of it is going to go there to the detrusor muscle. And another branch is going to come down here to the internal urethral sphincter. I hope you can see the conundrum. By trying to not void the bladder, I want to relax the detrusor muscle. At the same time, I want to contract my internal urethral sphincter. This is why I have different receptors, because I know what's being delivered. This is norepinephrine from this postganglionic, right? But now we can start to predict, because I want this to be inhibitory, because I want the muscle to relax, I'm thinking that this is going to be an even-numbered receptor, so either an alpha 2 or a beta 2. Now, I can go look it up in the table, and I'm going to see that 80% of the time I'm right. But down here on the internal urethral sphincter, what would I expect? Because I want this to be stimulatory, I want this smooth muscle to constrict so that I can block the exit of the urine, then I would expect this to be an odd-numbered receptor, either an alpha 1 or a beta 1 or a beta 3. And we would expect to see just the opposite for the parasympathetics, right? So because this is all about rest and digest, of course, we do see some differences where the sympathetic system, this is a long post-ganglionic, the parasympathetic is a long pre-ganglionic, we'll still branch off, and we're going to synapse onto a short post-ganglionic that's right here at the target tissue. But because the parasympathetic is all about voiding the bladder, right? This is rest and digest, and I have to go to the bathroom. I want this now to be excitatory. And so this is going to be one of my odd-numbered muscarinics, M1, M3, or M5. Whereas if we follow this one, synapse here at a collateral ganglia, a terminal ganglia, with a short postganglionic, this one I want to be inhibitory so that I can relax that muscle and allow the bladder to void itself. This is going to be an even-numbered receptor. So that's the idea. As long as we can visualize, do I want smooth muscle to relax? Do I want it to contract? And then can I correlate that? How do I do that? With a specific receptor type, and typically the evens are going to be inhibitory and the odds are going to be stimulatory. So hopefully that gives you a decent idea of how these two systems play off of each other to control the functionality of all these visceral organs.